Okay, here we go. Okay, I welcome all of you. Thank you very much for joining this class. I welcome all of you to for your molecular biology class, and today we will going to study more about that how the expression of a gene is controlled, and we will see and we will try to understand the details at a molecular level about the mechanisms and the modes of regulation of gene expression. It's supposed to be like everything for life sciences. When I said everything, that means that life is more about the control. It's more about that how well you can regulate the expression of a certain gene in a different in a certain under certain circumstances. As we all know that the environment in which we are living is constantly changing. With every now and then, we are receiving different types of stimulus, different types of external stimulus, different types of external stimuli. Sorry, and we need to respond to those stimuli according to the circumstances, and that is only possible if life is able to receive those signals, and if life is able to interpret those signals, and finally if life have some means to respond to those signals. So that is the only way that a system can exist as a living system. And the key, or I must say, the secret of life is hidden in the ability of life to control the expression of different genes in different circumstances. That is the only way that a cell can utilize its genetic information to execute different functions. The story is a little bit more complicated than we can assume. Let me make it simpler for you at the start. Then let's suppose if there is 100 different genes out there in a living system. So we might be thinking about having 100 different functions because we are carrying 100 different genes. That makes the things quite simple. 100 genes, 100 functions. But think about if we know how to tune up and how to tune down all these 100 genes in different ways. So in that case, we will be having several 100,000 combinations out of the expression of just 100 genes. That is what control is all about. So the versatility and diversity of function arises out of our ability to control the expression of the genes in different ways. It's about, let me make it more simpler before I go to my slides, that without the control, we have two options in controlling the speed of the fan. Either the fan is turned on or it is turned off. That's all. But if we have a control or if we have a regulator, we can guess we have all possible, we can control the speed of that fan, and we can have all those functions which were not there before the regulator. There might be two states. The one is the darkness and the other is light. But with the control and regulation, we can have lots of different shades of lights which exist between darkness and light. Okay, so let me start our formal lecture with this just little opening thoughts that how important is the concept of regulation and how important is the ability of our cells to control the expression of different genes. We will go and we will try to go into details of those mechanisms. But before the start, I want you people to think that there are certain genes which are not required to be regulated, which are not supposed to be regulated. Those are the genes which are required to be expressed under all circumstances. Whether a cell is type A, whether a cell is of type B, whether a cell is small, whether a cell is a large, whether a cell is doing a function A, whether a cell is involved in doing a function B. So whatever the type of that cell, there are certain genes which are required to be expressed in all circumstances. Those genes are involved into certain critical functions. And those critical functions are related to the energy production, are related to macromolecule synthesis, they are related to garbage disposal, and they are related to avoid the death. So those are the genes without them, no life is able to sustain itself. Those are called as housekeeping genes. For example, the genes which are producing the enzymes which are carrying out the metabolic pathway involved in respiration, all those genes are housekeeping genes. Whether a cell is small or is large, whether the cell belongs to type A, type B, or type C, whether it is executing a function called A, or whether it is executing another function called B, under all circumstances, the cell needs energy for its very existence. And that energy is created out of the very famous metabolic pathway which we call as respiration. And you know, it includes glycolysis, it includes Krebs cycle, it includes electron transport chain. So whatever the enzymes which are required to execute those metabolic pathways, those are included into housekeeping genes. And they are not supposed to be regulated. We don't want them to be regulated because we want a cell should always be able to produce energy under all circumstances. Under all circumstances. Similarly, there are certain other cells which are supposed to be involved in the function of Golgi complex. Golgi complex, you might remember, is a very famous organelle, and it is actually more important and more interesting than we originally studied about the Golgi complex. You can recall, you can recall your earlier biological understanding about biology, that there are some structures within the cells. I'm sure that you should be able to see. If not, then I will going to use another things. You might remember that the Golgi complex, or what you call them, the Golgi bodies, these are the structures which are responsible to produce certain packages, and these packages are created in the form of sacs, and those sacs are exported outside the cells. Anything which is required to be transported outside the cell is created maybe in the endoplasmic reticulum. It might be the proteins, and then transported into the Golgi complex. Golgi complex package those materials and then export it out. That's the one famous function you can recall. But this is not what I want you to ponder at this particular time. There are other important things. There are other important functions Golgi complex is doing. And that is to identify the unwanted material within the cell. To identify unwanted material within the cell. मैंने भी test मारा है बस. I just tested if the other camera is working or not. Okay, don't worry. मैं इसको बाद में use करता हूँ. So the concept, the Golgi complex is involved in identifying the unwanted material, and that is so critical. It is equally important like protein synthesis. You can recall your concept about protein synthesis, that every now and then the cell is planning to create different types of proteins to execute certain functions. But after a certain period of time, when the function is no longer required, you should think the destiny of such proteins. इन proteins के साथ क्या होएगा? Who have executed their function. If a protein is not required within the cell, or the function is not required within the cell, that protein must be degraded. That protein must be consumed or recycled. If a lipid is created by the cell and is no longer required, if a macromolecular structure is created within the cell to execute a certain function and now it is no longer required, that is also the job of Golgi complex and associated organelles, to capture those things, to destroy. Destroy is not a very good word. To recycle those materials or export them if they don't have capacity to recycle those materials, to export them as a garbage. So that is a very critical function. Think, if the ability of the cell, the recycling ability of the cell is hampered, do not work in a normal way, the cell will going to accumulate lots of unwanted material, and that will ended up in the death of that cell. That will going to trigger the death of that cell. So the recycling of unwanted material is so critical that the genes which are involved in the recycling of material or garbage disposal, they are also considered as housekeeping genes. So all those genes, whether they are like housekeeping genes, all the housekeeping genes, they are supposed to be, they are supposed to allow, they are supposed to stay active in all sorts of cells under all circumstances. So there should be no check and balance. There should be no, I must say, regulatory switch present in those genes. Those genes are called as housekeeping genes under whatever the circumstances. We don't want those cells to be regulated. We want them to stay active. The most easiest of the way you can relate housekeeping genes in terms of regulation, that the housekeeping genes should be present in an open portion of our chromatin. It should be present in active part of our chromosomes. And those active parts are called as euchromatin. It should be present in the euchromatin. There's the first thing. And further, you want their promoter regions to stay available all the time under all circumstances for the recruitment of RNA pol 2 and for the recruitment of transcriptional machinery. So you don't want any DNA methylation to be there in the promoter region of housekeeping genes. And actually the housekeeping genes are supposed to be a parameter with the help of which we quantify how many cells are out there, how much living activity and activity related to life is out there. So with the help of the expression of housekeeping genes, we can figure it out how many cells are out there. Because if you have 10 different cells, and that's an important concept you should understand, and this is important thing, that how the housekeeping genes help us to compare and to understand that how much life is out there. If you are carrying two different types of genes, two different types of samples, this is a one type of samples, you can consider it as a normal cell. There is another sort of cells out there, and you can consider these cells as bad cells or cancer cells. Actually, I should say another type of cells, but let's make it and use it as an example. So this is like a normal cell, and these are the cancer cells. The normal cells are carrying the housekeeping genes. The answer is yes. The cancer cells are carrying the housekeeping genes. The answer is yes. Both of them are carrying the housekeeping genes. If I ask you the level of expression of a housekeeping gene, let's name a housekeeping gene. Let's name one housekeeping gene as an X. It's a housekeeping gene. If I ask you, there are 4 cells out there in the normal cells and the 4 cells out there in the cancer sample. If the level of expression of a housekeeping gene called X is 10 here, it should be 10 here in the cancer cell as well. The level of expression of housekeeping gene will almost similar irrespective of the nature of the cells. But let's suppose there is a tumor suppressor gene, I'm sure you people can guess. The tumor suppressor genes are the ones, which can control the cell division. It actually slows down the cell division. The name of the tumor suppressor gene is G, let's suppose. If its expression is 6 points here, it will be less in the cancer cells. If the tumor suppressor gene is expressing with 6 points here in the normal cells, it is expressing with 3 points in the cancer cells. The expressions of other genes are different in these two cells, but the expression of a housekeeping gene will always close to each other. This difference, the difference in the expression pattern of other genes, defines the outcome of the cells, whether these cells will be like a cancer cells or these cells will be like the normal cells. If we have a cell type which is called as A, another cell type which is called as B, another cell type which is called as C, the housekeeping gene, the expression of housekeeping gene, if you are taking the similar number of cells here, if you are taking 10 cells here and the 10 cells of the B and the 10 cells of the C, you will get an equal expression of housekeeping genes, equal levels of expression of house, and that makes sense. But for any other cell, for any other gene which is not a housekeeping gene, A, it may be B, it may be C, it may be D, whatever, they have different expression in different cell types. And that is why different cells behave in different way. So the expression pattern of housekeeping genes will almost same if you are taking equal number of cells irrespective of the cell type. But in case of the other genes which are not housekeeping genes, their expression and their expression quantity, their expression values will be different in different cells depending on the nature of that cell. Let me share another thing with you. For example, the epithelial cell of the skin and the cardiac muscle cell of the heart, both of the cells are supposed to be the human cells. If you take equal number of epithelial cells from the skin and equal number of cardiac muscle cells from the heart, they will give you an equal expression of housekeeping genes. But you will see more genes which are related to the function of the skin will be expressed in the epithelial cells. And at the same time, you will see less genes are expressed. Those genes will not express which are related to the function of the heart. Those genes will not be expressed in the epithelial cells. So that is how the difference in the expression patterns of different genes define the outcome or the final phenotype of the cell. That whether this cell will going to act as a epithelial cell or this cell will going to be like a cardiac muscle cells. Here are some of the examples of the housekeeping genes. And as an MPhil student, you should know about uh, at least the name of those housekeeping genes, some important housekeeping. I'm expecting you to remember 18S, ribosomal RNA gene. I want you to remember ACTB, which is actin. Actually, it's a beta actin. I want you to remember GAPDH, famously called as GAPDH. This is another housekeeping gene. And most of you, many of you will going to use actin and GAPDH, expression of actin and a GAPDH to normalize the expression of other genes. It's a little bit complicated to understand at this level, but just remember, you need to calculate the expression of housekeeping genes first before analyzing the expression of other genes. I wish you should be able to understand this thing. This is called normalization. If two samples are showing same expression of housekeeping gene, that means these two samples are equal. That means these two samples contains equal amount of life. These two samples contain equal number of cells. If they are showing an equal expression of housekeeping gene, only then, after then, you are allowed to analyze the expression of other genes. Only after that, you will be able to compare these two samples. So if you want to compare the expression of the two samples, first you need to make sure that these two samples are equal. And there is only one way biologically at a molecular level that you can use to prove that these two samples are equal. When these two samples are showing an equal expression of a housekeeping gene. Okay, the people who will work on the expression analysis of certain genes, this will going to be their core concept, their core understanding about expression of different genes. I will talk more about these things maybe in the next semester if we get a chance, about how we proceed with the quantification of genes. Okay, so we all know that all the cells in our human body, they contains identical DNA. At least in my body, all of the cells came from the one cell. So a very important question is that why hepatocytes? Hepatocytes means the cells from the liver. Why the hepatocytes? They produce factor 8 and not the insulin. They're not supposed to produce insulin. They are supposed to produce the factor 8. Or similarly, you can ask another question, that why pancreatic beta cells, they produce insulin, but not the factor 8. Although both of them originated from the same cell and they are carrying identical DNA. It's a very important question. So that's how it is controlled and regulated. How expression of a certain gene is controlled and what are the factors which are present on the DNA or outside the DNA, which is responsible for the regulation of those genes. Some factors are present besides the gene on the same chromosome. Those are called as cis-acting regulators or cis-acting components or cis-acting DNA sequences. Some factors are present on the other chromosomes or on the or far away from the genes. Those are called as trans-acting components or trans-acting regulators, or maybe they are trans-acting proteins. So in the next part of the lecture, we will going to study some of the cis-acting regulators which can control the expression of a certain gene. And please remember that the cis regulatory elements are present besides the gene on the same chromosome, on the same stretch of DNA. Okay, so I'm stopping the share. So if you have any question, you can ask me right now. Okay, before I proceed to the next part. G.